Hi. So thank you everyone so much. So many thanks. Thank you everyone coming now. Thank you all the speakers on the panel. What we're going to try and do in this session is just muse and think and answer questions. Well, think about questions we probably don't know all the answers to. So please do in the Q&A pose questions, or maybe now is a good time to also use the discussion forum for kind of your points, and we'll read them out as well to kind of contribute them to the discussion. We'd really like to hear from the floor as well as the speakers. What we want to do here, I suppose, is reflect on the various different kind of themes, consistent themes that have emerged, and the kind of contradictions across the meeting, and think how we can sort of what we know now that we didn't know 2 days ago, and what we think we should be trying to do more of in the future, whether that's from a physiological research perspective or interfacing the clinic. So yeah, Andrew Murray's co-chairing this. So between us, we're going to kind of ask people questions and ask you on the floor in the audience questions as well, and see what comes out of the discussion really. So I was just going to start by noting, there've been so many different themes to kind of summarize. I think a really nice consistent thing has been obviously the occurrence of hyperventilation in patients, and I'd be really interested to hear why people think that happens. And I was going to particularly ask Oliver, because I think we've also heard lots of mechanism sort of angles as well, and like whether any of that can inform how we understand sort of hyperventilation features of that you see in long COVID patients. So what do you think, Oliver?
Yeah, really great question. And I think if you'd have asked me this 36 hours ago, I'd have said very confidently there's a central mechanism that links in dysautonomia and inappropriate hyperventilation. Done, answered. However, over the last day and a half, it's been really wonderful to have my eyes open to so many different potential mechanisms there. And what's been fascinating is this commonality that a few different groups working separately with different methods have found the same thing. So there is definitely an avenue for further research to understand that better. And now we've got a good target to look at what we're doing and the testing that we're doing, and think about other testing that perhaps might be good to tailor towards that. And branching out in the groups that we're working in, I know that there's been a conversation in the past about the groups that have been brought together have been quite siloed and perhaps not as much diversity in them between, a clinical team and a research team. And so in our team, we've got a physiologist, a biomechanic, and lots of different specialities of doctor, but not many kind of specific neuro specialists. So I'm looking forward to rewatching some of the talks, which is most unlike me to watch any talk a second time, but to pick out the bits that have really piqued my interest, and I'd be fascinated if anyone's got any potential mechanisms that they feel who are in the audience at the moment, because using the hive brain in forums such as this is fantastic, and a real credit to the Physiological Society for bringing these 2 days together, because they've been fascinating for me, and I think based on the feedback, a lot of the audience as well.
Thanks, Oliver. Does anyone else have any sort of things to add to that? How do we think? I don't know. How does? Yeah. Anyone else want to add to that? We probably. Oh, okay. Fair enough.
I guess
Sorry, go on.
And I was just going to say, one of the joys of the meeting has, okay, it's a strength and a weakness, but the fact that you've got so many different experts in so many different areas, it's it's really interesting to come in as somebody who's not particularly in any of the areas, because you see so many echoes of things you do. So when I was listening to Oliver's talk yesterday on breathlessness, it was me who posted the question about, well, what lung volume are people breathing at? Because you don't actually have to change that lung volume very much in order to change the work of breathing the afferents from the lung, the hemodynamics in the lung. And that comes from immersing people in water and diving. So it's a real strength to have this cross-disciplinary meeting where you can cross-fertilize. And actually the trend has been to go against that kind of meeting. But I think this has been a really useful multidisciplinary meeting in that respect.
Thanks. Andrew, you were going to say something.
No, I mean, I guess the analogy of the blind man feeling their way around the elephant and trying to work out what it is. I mean, it's something we all do. We work within our silos. We're often working on much more established conditions where there is a broader literature to draw on. But here, everything, you've got experts from all different areas looking at the elephant and finding differences. So where do we begin with this, and how do we avoid working within these silos in the future? I think that's going to be a big sort of practical question. I guess my comment in the chat agree it's an integrated physiology problem, definitely. I mean, I don't think anybody on this call will disagree with that. If you do disagree, post that in the chat by all means. But
how do we move? Sorry.
Go ahead.
No. Sorry, no. On that though, I was just going to ask, like, whether people think, how do we do that practically? In order to capture the different things that can go wrong, that different people have presented on needing finer grained approaches to various different aspects of long COVID. So whether that was Melanie Dunny and Boom Lim on the sort of blood pressure fluctuations that you need to like look at in more depth to notice them, or whether that's like mitochondrial dysfunction. What do we need to be incorporating into kind of what tests are done on people who present, or like whether it's like exercise tests or whatever, like how can we better identify or stratify patients? Because do you think in a practical, and also as Epidemiology says, that it can be rolled out to lots of people because it's difficult, I guess. Do you think? Does anyone want to speak to that? Or
I think just one thing I would say that is quite unique about this is that there's actually an appetite from the people with long COVID to find a solution as well. So there's willingness on both parts. Whereas very often we obviously struggle to get hold of people to try and address these questions. Here we've got a ready pool of people who will jump at any opportunity. So I think this is a time when we can really demonstrate what integrative physiology can do and really highlight its utility to the public. And then hopefully that will roll out on a much wider scale across a number of different diseases and conditions when we eventually solve this one, hopefully on a positive note.
I think from working in the ME/CFS space, this meeting's been really great because people are really assessing these people properly, doing lots of testing. And what we failed over a number of years in ME/CFS is doing the proper assessment. So a lot of people have gone into these studies that weren't ME patients. And so I think you've got the opportunity to make sure that everybody that comes into a long COVID study has long COVID, because there will be people that have long COVID effects that may not have, they may be suffering from something else. It's trying to tease those out. And if we can do that, then you get a much more homogeneous group. Because, for example, in the immunology space, this dysfunctional immunology, it's not really planned out. Some groups find something in some cytokines, others don't. And there's all these differences that you get with symptomology as well. So doing longitudinal studies, I think, is really important in terms of trying to establish things long term, because in the ME/CFS patients, they change. So metabolism changes, the immune markers change over time, and it's trying to get a handle on that. It's really quite challenging.
How similar do you think the populations are? And do you think there was one of the talks yesterday saying that though people started out with quite a variety of symptoms, they kind of became more homogenous as a population over time? And I wonder how, is that converging to something that looks more like ME/CFS, do you think?
I suppose it depends how many people you're going to be left with at the end. It's interesting, the Epstein-Barr glandular fever, the numbers are very similar, at 1% of people would have it long term. It's whether people do continue to get better, or do some people reach a point 2 or 3 years down the line where they've stopped improving, and they're the people that we capture with ME, because it takes years to be diagnosed. So you're getting these really quite late stage patients that come through, whereas with this group it's going to be different. There's going to be a mixture there. There would be people that are recovering very slowly that might be different. So I think it's going to be interesting to look really and see that. But and in terms of autonomic function, we did some work with a Polish group, and so there's a sympathetic driven group, and then there's the ones that aren't in the ME cohort when you start looking. But I don't know, was the impression from this group that they're mainly sympathetic drivers going on? I got that feel from the presentations today. I don't know who was
certainly the conclusion of some people. Yeah,
yeah.
It's certainly a consistent theme, isn't it? And Carl, could I come back to you? I mean, on our particular silo, if I can be indulged for a minute on mitochondrial. I'm not sure I'm happy with the term dysfunction or mitochondrial impairments. I mean, we don't know. I mean, the mitochondria are probably not the root cause of this, so dysfunction is probably not the right word. But do we know what's causing that? I mean, we've heard several different talks hinting at it, and is it oxygen supply to the mitochondria? Is it? I mean, you've talked about substrate flux through the mitochondria being an issue. We've seen interactions highlighted between the ACE pathway and mitochondria. What's your current thinking?
I think from the ME space, it seems to be the substrates. See, when you run these seahorse assays, you're not looking at mitochondria, it's all the other bits and pieces that go into them. So you could have a mitochondrial effect on a standard seahorse profile, but it could be the glucose supply. But these are patients, aren't they? So it's quite different from a PBMC experiment. I think the work that Betty Rahman's doing, looking at the changes in the phosphocreatine, is going to be really interesting to see what that shows. We did some work with Brooks a few years ago looking at lactate spikes. So they went up, but they took a long time to come down in the patients. So does that mean they can't get rid of it very fast? So is the brain not using it up as a fuel? So, but I don't know whether we can't really say. I get the impression the group in Newcastle looked at the individual complexes in the patients, and there wasn't a problem with the individual components of the mitochondria. They were there, they were functioning individually, but there's something going wrong with how it's all put together. I think this sort of pseudo hypoxia idea is an interesting one around, maybe there's a poor supply of oxygen. Maybe that has the knock-on effect. But we've never looked at the right tissues. We need to look at the muscle and the brain, perhaps with these non-invasive methods. But we couldn't never get ethics to do a biopsy in a patient with ME, but I hear that you can do that in the long COVID group. So maybe that will shed some light onto this.
Yeah, and you highlighted the pyruvate dehydrogenase inhibition, which is an established downstream target of the hypoxia-inducible factors. So yes, I mean, it could be downstream of microcirculatory dysfunction and
yeah, and the work that Damien Tyler's doing in Oxford with the sort of DNP that's now gone into cardiac work and also in cancer, if you could use that type of approach to see where does the pyruvate go? Does it go into the mitochondria or out to lactate? That would be really informative. These studies cost £5,000 a scan. So, but if we can get the budgets to do that, I think that's what we need to do to add to the work with the spectroscopy that Betty's doing.
Yeah, no, I agree.
So to take it sort of more back to kind of epidemiological perspective, like where we need to be going from a kind of linking, the physiology with a sort of broader cohorts of people. Do you think, Nisrin, could you maybe comment on sort of what we need to be recording maybe going forwards as we stop recording anything, how we sort of deal with that transition?
I was going to say, because obviously cohorts were mentioned, and I think my observation from an epidemiological point of view is that there could be a lot of bias in the study, even physiology, pathology in that study introduced early on by defining who has long COVID and who doesn't right from the start. So if you're studying cohort patients, you have to have some way of defining long COVID and then some way of defining your control in many studies where you're comparing long COVID and to other. So either COVID recovered or no COVID usually is the control. And this is really tricky because we've got no good way of defining long COVID. So when you talk about having a homogeneous group, you can maybe even get to that by introducing lots of bias at the start of the cohort, depending on how you're defining the group which has long COVID in. And then the findings would just reflect that largely rather than reflect kind of the reality of people with long COVID. I'm not proposing it that there are no easy solutions, because there is no way to establish what is pure long COVID to study now. We haven't got that test.
Is there a pure long COVID? Do you think that's a thing?
Whatever you want to call it, but I do think there is a pattern. I do think there is I think there's wide variation in severity. And what's also happening is that there are people who have long COVID, maybe managing their condition much better, and they're not necessarily being classed as kind of the severe end, or not necessarily even the severe end. It's about, a lot of it is about resources and what your life allows you to manage. I've experienced long COVID. If I was in a job where I had to kind of run around or stand or lift things all day, I would just be completely out of job for the last 2 years. But, I've got the resource to actually be able to be flexible in terms of my job to accommodate for my condition. And then I'm functioning near normal. And I think all of these things are really important to consider, even when you're looking at the physiology, pathology, because it's about who you're looking at and what sort of subset of patients or people you're looking at, and how they're being selected. So I think that's important. It's kind of the start of who you're studying and how you define. We've seen a lot of epidemiological studies where there's obvious misclassification bias, huge at the start, because there's so much mixing in terms of, we think you've had COVID. You think you've had COVID and you covered. We think we've got long COVID, and we've used things like PCR test or an antibody test, where there's a lot of limitations around them. There's a lot of criticism around in my discipline, and this is something I've studied for a while. We always think whatever is self-reported is weak, and you need to assess things objectively. This is a higher way of assessing a more reliable. Actually, with long COVID, I don't think that necessarily applies, because we haven't got that good objective way of assessing it. So until we get there, we have to get much better at classification using reporting as much as we can, and that's basically by clustering people across different symptoms, but also across the kind of the severity and across their characteristics and their resources as well, in terms of how they're managing their clinical condition. If that makes sense.
Well, it's all data, isn't it? We just always need more data about, or just to be able to describe exactly where you fit on a symptom spectrum and who you are and what variant of COVID you might have had, and we'll put that all together.
Yeah, but we have to acknowledge the people that there's no data. That's the thing. The data is largely skewed. If you look at healthcare data in terms of who gets the diagnosis of long COVID, because I'm working with that. It's just very, very much skewed. We know that, who gets that label of long COVID. But even epidemiological data is skewed, and that's something that in whichever sample you're drawing from, I think that's something really need to pay attention to, is that where you're drawing that sample from.
And presumably Liz, you can maybe speak to this, because obviously you said in your talk, acknowledging it's a very skewed sample, but then, that plus healthcare data plus, do we start to get somewhere by looking at multiple sources of, for example, the hyperventilation result is nice because it's there in lots of different studies with different groups, and it seems nice and consistent. So we can be confident that's there. But could you speak to that maybe?
Yeah, we're existing in a space where there's not a kind of nice biomarker that correlates perfectly with the phenotype of long COVID, don't we? So I think everything Nazreen said is absolutely correct, isn't it? And we're working often, as I think a lot of the reflections, especially in my session, were just how quickly our understanding of the condition has changed at each moment, and how many new studies we could plan as kind of the pandemic has evolved. And I think that's true with long COVID. Our understanding of long COVID has evolved. Our understanding of the phenotype, the trajectory, who's at risk, it's still evolving, and it's difficult in that space to even produce research that's kind of comparable one study to another, because what researchers did at the time was their best estimate of what they should measure and how they should define the condition, and then things changed. I think that's coming back to that point that preprints have been so important. It has been so important to get results out quickly, hasn't it? The other thing I've seen that I find really exciting, especially in this meeting, when I was working with long COVID clinic clinicians, as soon as they were set up, so over a year ago, it was all focused on kind of lifestyle changes and pacing and everything, as if, you know, there wasn't going to be anything physiological that could be done for these patients. It was going to be about pacing and waiting to get better. And I think this physiological research that's coming through that shows, you know, clear biological things that are different in long COVID patients is exciting. And I think the dissemination and communication of those findings from research physiologists to clinicians is going to be really key in terms of mobilizing resources to implement findings and do trials of new therapies. And I'm really hopeful that this could spill over for the ME population as well, who are just chronically under-researched, and, you know, finding physiological components to that could be treated is going to be really important as well. So I hope that will come out of the long COVID research as well.
One question Catherine and I were throwing around after, I mean, particularly after Danny Altman's talk yesterday, where he really didn't want to be pinned down on different immune subtypes and said measure everything. I mean, we've got the capacity now to measure, you know, plasma metabolome, tissue metabolomes, proteomes, you know, the full inflammatory profile. I mean, we can throw the big data sink at this and really generate a lot of data. We've got a lot of patients we could work with on this. How useful is that in comparison with the kind of studies we've heard about, where people have been looking at individual mechanisms, their own sort of pet area, and seeing clear differences? But you know, how useful is big data going to be in addressing the long COVID problem? I don't know who wants to take that on. Anybody willing to give it a go?
I'm happy to say something again, but I mean, I think there's a lot of potential, but it's going to require a lot of joined up thinking. You know, with big data analysis, it's, you know, garbage in, garbage out. So you need to be sure of what you're looking at, how you're analyzing it. And of course, with, you know, just churning through data, there's always the chance of turning up spurious results. So you need to have a good pipeline for like taking potential leads and then having a good way of investigating their kind of validity within the patient group.
I don't know much about the immune cell assays, but are we going to get a functional readout of what the cells are doing, or is it just going to be the numbers of cells in sort of these cytoph type approaches? Or what's the output's going to be? There was lots of proteomics stuff yesterday, so from our own work on metabolomics, it's been quite frustrating because, you know, we do untargeted metabolomics, and there's so many things there that we don't know what they are, and it's really difficult to, you know, you can't pin them down to pathways. We know they're different, but it's trying to do more studies. Put more, it needs more money invested to find out what these things are that are very different across the groups. What about you, Andrew, from your stuff?
Well, no, I mean, I agree. I mean, you know, with the untargeted approaches can be great for hypothesis generation, but you know, with our work in, say, hypoxia in the ICU patients, we've gone in with a more targeted approach, looking at, you know, pathways of interest that clearly link with mitochondrial function and energetics. So that's been easier to get our head around. The open profiling is, you know, you get some fascinating hits out of that, but I'm not sure. You know, do we have the time to do this? I mean, this is an urgent problem, so it's.
But don't we need to do both? I mean, one of the comments on the treatment session actually was a slightly frustrated, or maybe rather very frustrated patient who was saying, you know, this is all a bit too little too late. We've already tried all the things. These are all the same things we've already tried. We need something different. And in a way, I suppose what the omics approach can do is shallowly sort of throw out things nobody's thinking about at the moment. And you then need the targeted approach, because obviously you don't then know what they're doing. They're just like, but I mean, naively, without, that's not what I do at all. But that seems like a sort of, I mean, that's not my area of research. It sort of seems like that would be a sensible sort of way of finding.
Yeah, I mean, absolutely. Yeah. No, go ahead. somebody was. No, I mean, I quite agree that both approaches are needed. I mean, in some ways, I was being provocative there, but I'd be, I mean, as physiologists, we could, throw big data at this. We could drill down into the mechanisms. We could spend 10 years trying to solve this. I mean, that's not helpful for the patients who are currently suffering with long COVID. So, I mean, are there quick targets? Are there quick things we can do as physiologists? I mean, I guess if we knew the answer to that, we'd already be doing them. But what should we be doing immediately? I mean, I'd be interested to get anybody's perspective on that, really.
We had a bit of a conversation about this earlier on, and about how, taking quite a pragmatic approach can be quite helpful. So managing the symptoms means that you are helping someone, and you're not just waiting for the solution to arrive, but trying to do things concurrently. So not just doing nothing and doing lots of investigations that lead to naught, but kind of trying to help and using different people's work. And what's been really good is watching how many people have come at it at a different angle. And one of the questions in the chat box is about physiological testing following exercise intervention or activity intervention. So, clearly different groups are doing different things, and we have been doing research alongside our clinical service with some of the individuals and some research participants. So trying to do everything for everyone is perhaps impossible, but trying to do as much as we can for as many as we can is perhaps achievable. And an integrated approach between a kind of big data versus individual, with some clustering and some cohorts and epidemiology, all of those things together, you can solve a problem quicker. But there's an incredible amount of data out there. Just every day you log on and there's another 5,000 papers to chew through. So that's been one of the struggles for us, kind of keeping abreast of what everybody else is doing.
Andrew, should I just add something to that? I mean, to some extent, the approach you adopt, I think, depends on where you are in the evolution of your understanding of a disease anyway. And I just give you an example, which may not be a parallel, but it seems to me it might be, which is non-freezing cold injury, where people end up with a range of symptoms. And people have approached this from different perspectives, very simply from a vascular perspective or a neural perspective. And the vascular groups say it's a vascular problem, and the neural groups say it's a neural problem. But actually what it probably is a neurovascular problem, with one preceding the other, or in some cases purely neural, in some cases purely vascular. I.e., you have to entertain the possibility that this is not one thing. It's one set of symptoms, but it's not one mechanistic pathological process, in which case you end up now saying, well, now you've got NFCI 1, NFCI 2, NFCI 3, and the first and most important thing you can do in doing the best for the patient is to work out what form they've got of that particular disease or that particular syndrome, and from there you can then plan a treatment regime. And I wonder if we'll get to that stage with long COVID, with several subcategories. But at the moment, it's so obscured by all the other variables that are going on. It's quite difficult to come to any conclusion on that.
I certainly see this with the mechanism when that's the start. So maybe a year ago, there was all these hypothesized mechanism, and the focus really was on kind of trying to define and characterize long COVID. Now you can see, which is, I think, is a bit of a sorry state, where there are more papers on the mechanisms, and there's almost like a competition on what is their mechanism of long COVID. And we know that there could be, is it persistent virus? Is this an autoimmunity? Is it mainly the clotting? And we do know these things can work together in a cycle. And I think people just need to talk a bit more to each other. People investigating these different mechanisms need to talk more about, because we need to have it focused on the person. And these are not exclusive mechanisms. We know that diseases. But actually what we're seeing is almost like saying, well, no, this is it, and we need to focus on this, and this is the ultimate solution, which is unlikely, really, if you focus on any one of these, kind of emerging mechanisms. I haven't managed, unfortunately, to be here for the whole of the 2 days, and I really look forward to kind of watching these talks if possible. But I'm hoping that provided more of an integrated way of proceeding. But that's the narrative, and I think the patient, you mentioned the patient, and I think it's very, it's frustrating as a patient to see these different proposals of mechanisms while they're actually in their position as a patient not being offered practically any kind of cure or treatment. So then they go and try and pursue understanding these different mechanisms, and in that pursuit they're saying, well, it's this or it's this, and it becomes very, very confusing, I think, and not very helpful. And I think any talk of the mechanism need to be always communicated in a way that this is a multi-system disease, in the vast majority of patients, and there is likely more than one underlying mechanisms in sequence or together, whatever, and it won't be just take this one drug for this one mechanism, certainly without any evidence of trials or anything, which we don't have at the moment.
Kind of following up on that point, I would be very much of the opinion that understanding the mechanisms is vital and very interesting, but is it ultimately good? How nuanced can we be in our approach to rehabilitation? Are we really going to be able to get down to the point of saying, right, this person is in this channel, therefore they need this one thing? Or ultimately, are we going to end up with a fairly broad spectrum rehabilitation strategy, and therefore some of our resources are perhaps more usefully focused, certainly in terms of those who have already experienced and continue to experience long COVID, in identifying the effective rehabilitation strategies while we continue in the background working out the mechanisms underpinning them. So perhaps as much as us physiologists, we're fascinated by understanding this, maybe we need to focus slightly more on the day-to-day of resolving it. And as I say, the other bit can continue in our own time effectively.
No, I was really struck by the 3 principles that Ami Banerjee alluded to in his talk, which is underpinning all of his Stimulate ICP work, which is the recognition, research, rehabilitation. And I mean, it strikes me that, these different research approaches are, obviously fascinating, hopefully important, but need to run alongside, what we can do to support the patients. First of all, what is currently available. And, we've heard various strategies that may provide short-term, perhaps lasting benefits. And similar to Melita's talk yesterday about inspiratory training, and hopefully that will continue to bear fruit. But recognizing that it's the patients who are at the forefront here. So
there's a couple of questions come in. I don't know if we've got anyone who has the expertise to answer this, but about whether or not the kind of the female differences in long COVID sort of frequency could be to do with the uterus being a sort of potential virus reservoir. And does anyone know anything about that? I'm not sure whether we'll have people who have that expertise here to answer it. But what I would encourage is if anyone in the audience does, please do also answer in there, because
there are some potential hormonal effects in terms of the microbiome. So there are some interesting difference between men and women that have been published in the literature. So maybe they are playing a role. But I don't know when people get to the menopause, do these all change? After the symptoms change, we don't really know.
I just want to stress what I said. I don't have an answer to this, I'm afraid, but and again, proposed mechanisms. I do want to stress from an epidemiological point of view that the better studies have not shown a massive difference between women and men in terms of prevalence. It's slightly more common in women. It's not massively more common in women. But I think there is obviously, again, bias in terms of, how you do the study, how you do the reporting. But actually, if you look at, say, the ONS long COVID estimates, the difference is quite small in terms of the prevalence between men and women, not big.
Good point, thank you.
So again, I was trying to say, if there is a mechanism, it might explain some of it, but certainly won't explain, kind of the main of long COVID, because it's common in men as well.
Yeah, no, sure. I mean, I think their point was that it was a potential additional virus reservoir, if that was an explanation, as the gut had been mentioned earlier in the day as a potential. So, yeah, thank you. And I think we've... So, I mean, some of the other questions, I mean, think about themes that have kind of come up, and I don't know how people want to talk about this more. I was kind of interested in what people think the dysautonomia role is, like how we can see dysautonomia as interacting with the vascular, sort of microvascular disorders and mitochondrial dysfunction. Like, can we see a sort of hypothesis for how you could get both those features with a common underlying cause, or do we think they're separate? My kind of, gut feelings, there must be, a limited number, maybe more than one, but limited number of kind of core, functions, features that cause other things in these different organs and systems. And obviously then, like Carl said, you might then get or was it Andrew? Anyway, sorry, but mitochondrial, not dysfunction. What do you say? Impairment. Sorry.
Impairment, I think, yeah.
It comes after something else, but then there must be some fewer number, one or a smaller number of things that kind of cause those different damages. What can be causing? What's likely to be causing dysautonomia? Is that, what level do we think from the different data that's been presented that could be happening peripherally, like in the brainstem? There was some discussion in the chat about whether sympathetic hyperactivation was evidenced or not, and someone cited a paper, but I'm afraid I haven't got. Can someone tell me what the outcome of that paper was, so that we can, has that been evidenced, or is that just speculation at this stage? I don't know if someone in the chat can but does anyone in the panel want to speculate on how those things might interact?
I don't know, this idea of brain stress or stress on the brain is quite interesting. What goes on in Oxford looking at the impact of stroke in different models. And you do get things coming out of the brain, linked to the brain, like vesicles come out, and they can turn off mitochondria. When animals hibernate, they can turn off mitochondria. So we have ways of turning off mitochondria naturally. So I sometimes wonder, is this involved somehow? When you get the full-blown symptoms of the flu, you really get tired. And is our mitochondria is shut down? Is that involved there? And we don't really understand what's going on. So how much of this is a virus or a pathogen, and how much of this is the host response? And just trying to work out which comes first, and how it all fits together is the challenge, really.
It's a really good question as well, because I was wondering, Paul Edison's talk right near the beginning of the meeting, where he was showing the differences in glucose metabolism in the brain. That was in acute COVID, I think, but it'd be really, really interesting to see what happens in long COVID in terms of like whole brain glucose metabolism. I don't know if people have done that or not, but that would seem to be.
So we haven't looked specifically at glucose, but we've got brain MRIs and neurofilament testing that we're going to analyze and compare to the symptoms and other phenotypes and the objective cognitive assessment, and see if there's any mechanistic hypothesis that fall out of that, which then we can kind of link in with other groups. So I'd hope to be able to partially answer your question at some point, but don't need a bigger brain than I to understand it all.
Structural, or are you going to do functional MRI?
So we did structural. We designed the study, quickly, as many people did. So we felt it was important to kind of get as much good stuff as we could, and then afterwards we were like, oh, it could have been, would have been nice if we'd done this and done that. So it's just structural, but we've added on some neurofilament work, and we hope that might help be able to close that gap in knowledge for us a bit.
So can you look at inflammation? There's some of the work in ME/CFS is trying to look at microglia activation, but there aren't very good markers of that. So they tend to look at lactate glycolysis as a surrogate. And is there anything, Oliver, that you could do to look directly at, is there inflammation in the brain of these people with long COVID that could be linked to the virus? Because I know that the NIH did quite a lengthy study looking for virus in all parts of the body, and they didn't find it really outside the lungs. when they looked in their studies, but are they using the right markers or tests? I don't know. Is anybody doing much in long COVID, looking at the brain?
I mean, there
I
must
don't
be.
know exactly the ones we're doing, so I can't answer your question directly, but I would hope in due course I'll be able to update you.
Yeah, there is one. So there's a TSPO is a mitochondrial component, and when you label that, you can look at microglial activation, but we don't do that in the UK. It's really difficult to do because I think you have to make the isotopes really close to where you are, and that would be a direct measure. But no one's really done it properly.
I mean, this is speaking from a position of ignorance, but there are supposed to be sequences, MRI sequences, that do allow you to see microglial like activation state. I think because the motility of the microglia changes, I assume, and so the diffusion characteristics of the brain change. But I mean, it's
there was a small study. So in the States, they were looking at lactate levels in the brain and heat temperature, and the temperature was going up, and they were saying that. But then it could all be uncoupling mitochondrial function. You don't really know what you're looking at in terms of the system.
Yeah. Hopefully Oliver's work and other people's will also help. But I mean, again, you still don't know, to be honest, if someone has decreased glucose metabolism and a brain area and they're feeling tired, what's cause and effect? I mean, you know, are you, you know, you think differently when you're tired, but is that the reason that you're tired? Probably not. I shouldn't say tired, I should say fatigued. I'm sorry. But.
Sure. Somebody mentioned ketogenic diets. So it's quite interesting. We did a small study with Oxford Brookes where we had ME patients and controls on ketogenic diets. And what was quite striking was that the controls hated it. They absolutely hated this diet. But the patients didn't really care. You know, they probably felt pretty bad anyway. But there was a real difference in their lactate changes after the exercise when they put them on the CPET. So on the ketogenic diet, there's some really strange differences in the traces. So they generate a lot of lactate, the ME patients, and where's that coming from? You know, they weren't using glucose. It was quite strikingly different. And then the ability to remove the lactate was much slower in the ME patients than the control groups are. So their lactate stayed high a long time. So again, it's pointing to some metabolic issue. And there's a study coming out of Maureen Hanson's group actually looking at the impact of 2 lots of exercise on metabolomics profiles. And they've I know that the paper's actually being reviewed at the moment, but there was they showed the data, and the big changes in metabolomics came in the interim period. So when the patients had done their first exercise, when they measured them at baseline before the second one, there was real differences. So this post-exertional malaise was really something you could measure using that approach, which, you know, getting this PEM assessment.
Could that be?
Did you say this is ME or long COVID?
No, ME. I don't think they've not done these studies in long COVID as far as I'm aware yet. So this is an ME cohort.
But that should really be tried in long COVID.
You could apply, yeah. Well, I suppose the challenge is putting these people through an exercise, because in ME they really don't like it, and also some of them can suffer for a long time. So it's trying to work out how you can put somebody through a milder sort of challenge. There's a cuff that the Canadian groups use, like a massage cuff, that seems to be able to induce lots of microRNA changes. But whether that's doing the same as a exercise bike is another factor. But yeah, it's how we challenge a patient that's really compromised with fatigue and all the rest of the things that they are, in a way to get some more information without really making them worse. That's the.
If I may just come in on the word fatigue, I really don't like it because it's just so vague. I mean, you said, "Oh, not tired fatigue." Yes, I mean it is not tiredness. It is something much more profound. But even the word fatigue, I think, even for the patients, it means different things, maybe to different people. And I really haven't seen a widespread good definition of what you are asking patients when you say, "Do you have fatigue?" You know, and that, I think that applies to long COVID as well. And I think we just need to get a bit better about kind of describing, asking it in more detail. And obviously there are scales for fatigue, so you can ask it in that way and ask different questions.
There's good ones in me. So there's something called the DIPPLE scale, came out of Chicago, but it's a really long protocol. A lot of patients go into a crash just doing the protocol. And but there are good to say, it's not just, as you say, it's not just fatigue, it's a whole bunch of stuff. It's more like exhaustion than fatigue, I think, in general.
In our survey, we asked about exhaustion just as a symptom, and then we did a fatigue severity scale, and that was both were very high in terms of prevalence, but the fatigue severity scale was higher. So when it's assessed by a scale, it could be as a direct question. But even exhaustion is not really a very good word. So it's just these things about and post-exertion malaise obviously is much more specific, but in long COVID as well, there's been, I personally prefer the term post-exertional symptom exacerbation rather than because the post-exertional bit, again, the bit which is the malaise, what is it? And it also depends on your set of symptoms that you experience, because then they could be exacerbated, but that's maybe the malaise for you, but they're not the same for everybody. I think, again, from an epidemiological point of view, and then when you're assessing cohorts, we really need to work a bit more on what we're asking in the questions and how we define things. I wonder if Anushka, is there anything you have any views about these things? Because I watched the video at the start, and you and also the other bit in long COVID, which is really not well characterized, is that sort of the course of the illness, the fluctuating and the relapsing, remitting, and the time points between, and all of that. It's just way oversimplifications when describing this in studies.
Yeah, and I think that's very true. So actually, on one day you might wake up feeling great, and by lunchtime feel rubbish. But also at different points in my disease now, which is just 5 months. So different symptoms have been of more importance to me at different stages. So, it was interesting to see some people presenting that 80% of people were saying that they've got fatigue, and that's sort of the biggest symptom. Well, at various points, actually, the fatigue hasn't been the issue for me. The fatigue has been the exertional tachycardia, or the sort of probably muscle fatigue, but the sort of shakiness in my muscles. And actually the fatigue was something I just thought, well, I could just deal with that. Now, as I feel like I might be getting closer to work, and sort of feeling that brain fog is gone, I now think, oh, actually the fatigue is going to be a big problem, because what am I going to do at 2 o'clock in the afternoon when I really need a nap? So actually the importance you put on it and how you experience it, it changes on a day-to-day basis, but over the course of your illness. And I don't know what the right term is. I, like everything, what one deaf word means to someone, it means something different to someone else. Heartburn, indigestion, whatever, chest pain. Everyone views it something slightly differently, and there's always a little bit of a discrepancy. But there do need to be some sort of standardized definitions, I suppose, really, so that we really know what we're dealing with.
I guess the functional assessment to me is the most important. Is that whatever symptoms you're experiencing, how it affects your functional ability, and that did make it into the WHO definition that impacts day-to-day activity. But it's worth noting that, say, in the ONS estimates, not everybody has that. About 2/3 say that their long COVID limits their day-to-day activity. So not everybody with long COVID will have that. But I do think, again, it's really not straightforward. It is complex, because what happens with long COVID, certainly what happened to me and some other people I know, is you really do adapt your life so you don't trigger your symptoms. So when you say it doesn't limit your day-to-day activity, it's because you've changed the way you do things as much as you can, so that it doesn't limit it.
Yeah, that we sort of adjust to how people have changed with how they've adapted as well, because that could change a load of physiological measures as well, I suppose. But what we'll have to do now is thank you so much, everyone, for so much brilliant conversation. I'd like to thank all the speakers, all the chairs, everyone who submitted abstracts and gave brilliant flash talks, everyone who's attended and contributed as well. But I'm going to pass over now to Mike Tipton, who's going to summarise his thoughts on the meeting and also introduce some kind of concepts and ideas from policy document he's been working with the Physiological Society on kind of how to take long COVID research and how to implement that in the future as well. So thank you very much, everyone, and I'd like to hand over to Mike. Thanks.
Thank you, Catherine. Yes, so I'm a trustee of the Physiological Society, and it gives me great pleasure to put some closing comments together about this meeting on long COVID, and what a meeting it's been. From the fascinating and thought-provoking first videos of Anusha and Mark having to deal with long COVID to the panel decision we've just had, the meeting, I think, has been of the highest caliber and a great example of what learned societies can achieve in bringing together different groups, working in different areas to improve knowledge and exchange and understanding. It's really been first class. So we've learned much. It's clear that long COVID is a widespread problem that is remaining below the radar in some critical quarters. Whilst we're learning more by the day, there's still much to learn if we're going to help the millions of people afflicted with this prolonged and in so many ways damaging illness. It's clear that long COVID has multiple morbidities and affects many physiological systems. We've heard of the excellent work and approaches to the immunological challenges. We have seen changes in respiratory function, gut function, autonomic function, microvascular, biochemical and cellular function, and psychophysiology. We've heard about different investigative methodologies and seen impressive evidence of neurological problems that may underlie organ damage, as well as the chronic pathology. We've heard what different groups, including the military and elite sport, are doing to assess, treat and rehabilitate patients. However, due to the multifaceted dynamic nature of long COVID and variations in individual susceptibilities to different pathologies, we remain unsure if a general pathology can be described or even exists. I think diseases such as long COVID test the very nature of the way we gather knowledge. At the end of this meeting, I'm left in little doubt that physiology has played a central part in the response to the acute phase of the COVID pandemic and will continue to be at the forefront of the fight against the long COVID pandemic. We have the expertise to dive deeply into different pathologies of long COVID, and we should do this. I think the bigger challenge is presented by multi-system diseases such as long COVID is how such essential and potentially disparate work is effectively and efficiently coordinated, funded and communicated. How different groups and disciplines are brought together quickly, provided with key data and enabled to address critical questions. And I think this is something we have to think more about going forward. Like many other organisations, the Physiological Society has worked hard over the last couple of years to support the fight against COVID-19, and it's learned and relearned many lessons along the way, including the need for collaboration. Indeed, much of what the Society has done in this area has been in collaboration with the Intensive Care Society, a recognition of the added value obtained by closely linking clinicians and physiologists. In addition to the research being conducted, the Society and its members must continue to advocate for a greater role for physiology in tackling unmet needs in patient care. With this in mind, I and the policy team at the Society have been working with the Intensive Care Society, along with the clinical and academic physiologists, to develop a short consensus paper, which outlines the following: the role of physiology throughout the pandemic, where and how physiology and physiological research could have been better and more rapidly deployed to support frontline workers, had structures existed to put this in place, how physiology can be embedded into decision-making at the top of government, how the Society can continue to work collaboratively with other learned and professional societies to build the networks that will be responsible for rapidly reacting to the next health emergency. This document's recommendations will aim to ensure that physiology fully informs the ongoing response to long COVID, as well as the world's preparedness for the next pandemic, making maximum use of the type of expertise we've heard from in the last couple of days. The document's only possible because of the quality of science that members have produced in response to the new health threat. As one of the steering group members notes, modern medicine is evidence-based, but in the absence of trial data, new diseases like COVID and long COVID are really addressed through experimental medicine with an understanding of physiological mechanisms at its core. The report steering group has made good progress with the document, but I would encourage anyone who has worked closely on demonstrating the value of physiology during the pandemic to get in touch with the team, as they're always keen to ensure that the contribution of physiology to answering the world's greatest healthcare challenges is recognised. The document will be launched shortly and we'll send more information to the attendees of this conference. Lastly, but certainly not least, I want to thank all of the organisers of the conference, especially Dr. Catherine Hall, who has, for her first conference as chairperson of the conference committee, has set the bar really high for herself. So keep that up going forward. Also, Professor Sue Dukas and Professor Andrew Murray, Professor Dan Martin, and the Physiological Society events team, Sarah Bundock, Caitlin Oates, and Becky Jones, for all their hard work and keeping the show on the road. Finally, thanks to all those who chaired and presented, and everyone who has attended as delegates for their time, enthusiasm, and involvement. I look forward to meeting you all again, hopefully in person, and hopefully very soon. Thanks very much. Happy to hand back to you, Catherine. You may be muted.
Oh gosh, what a way to end. Just, I was always saying was thank you very much. And also, I think that's it really. Just thank you for coming and attending. And it's been really fascinating and given us such a lot to think about going forwards. So yeah, thank you for your time. And oh, and I meant to say one last thanks. Emily Wiley, who made the video that we saw at the very beginning, thank you to her as well, from the Physiological Society, who put that together, because I think that was such a great way to start, and I forgot to thank her at the time because I'm terrible. Anyway, so thank you again and goodbye.